two. 1, 2, 3, go! Wonder Peeps, nandito na. Magpe-perform sa inyong harapan sama-sama. Hindi malalaki at hindi malalakas, pero pag nagtulungan, lahat malulutas. Go Wonder Peeps! Yay! Amen!
Hi guys, this is the Wonder Peeps, and in this video, we are gonna talk about cellular respiration. Cellular respiration is a set of metabolic reactions and processes that take place in the cells of organisms to convert biochemical energy from nutrients into adenosine triphosphate or ATP and then release waste products. Cellular respiration has 3 steps: the glycolysis, Krebs cycle, and the electron transport chain. Glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. Glycolysis consists of an energy-requiring phase followed by an energy-releasing phase. Glycolysis is a series of reactions that extract energy from glucose by splitting it into 2,3 carbon molecules called pyruvates. Glycolysis doesn't require oxygen, and many anaerobic organisms or organisms that do not use oxygen also have this pathway. Glycolysis takes place in the cytosol of a cell, and it can be broken down into 2 main phases: the energy-requiring phase and the energy-releasing phase. In the energy-requiring phase, 2 ATP are spent to form an unstable sugar with 2 phosphate groups, which then splits to form 3 carbon molecules that are isomers of each other. A phosphate group is transferred from ATP to glucose, making glucose 6-phosphate. Glucose 6-phosphate is more reactive than glucose, and the addition of the phosphate also traps glucose inside the cell, since glucose with the phosphate can't readily cross the membrane. Step 2. Glucose 6-phosphate is converted into its isomer, fructose 6-phosphate. In step 3, a phosphate group is transferred from ATP to fructose 6-phosphate, producing fructose 1,6-biphosphate. This step is catalyzed by the enzyme phosphofructinase, which can be regulated to speed up or slow down the glycolysis pathway. In step 4, fructose 1,6-biphosphate splits to form 2 3-carbon sugars, dihydrocyacetone phosphate or the DHAP, and glyceraldehyde 3-phosphate. They are isomers of each other, but only one glyceraldehyde 3-phosphate can directly continue through the next steps of glycolysis. Next is step 5. DHAP is converted into glyceraldehyde 3-phosphate. The 2 molecules exist in equilibrium, but the equilibrium is pulled strongly downward in the scheme of the diagram above as glyceraldehyde 3-phosphate is used up. Thus, all of the DHAP is eventually converted. The second step is the energy-releasing phase. In the second half of glycolysis, the 3 carbon sugars formed in the first half of the process go through a series of additional transformations, ultimately turning into pyruvate. In the process, 4 ATP molecules are produced along with 2 molecules of NADH. In step 6 of the glycolysis process, 2 half reactions occur simultaneously. First, the glyceraldehyde 3-phosphate, which is one of the 3 carbon sugars formed in the initial phase, is oxidized, and second, NAD is reduced to NADH. The overall reaction is exergonic, releasing energy that is then used to phosphorylate the molecule, forming 1,3-biphosphoglycerate. In step 7, 1,3-biphosphoglycerate donates one of its phosphate groups to ADP, making a molecule ATP and turning into 3-phosphoglycerate in the process. In step 8, 3-phosphoglycerate is converted into its isomer, the 2-phosphoglycerate. In step 9, 2-phosphoglycerate loses a molecule of water, becoming phosphoenol pyruvate, which is an unstable molecule poised to lose its phosphate group in the final step of glycolysis.
Ay, nagugutom na naman ako.
In the last step, PEP readily donates its phosphate group to ADP, making a second molecule of ATP. As it loses its phosphate, PEP is converted into pyruvate, which is the end product of glycolysis.
The second process of the cellular respiration is the Krebs cycle. The Krebs cycle is named after its discoverer, Hans Krebs.
The Krebs cycle is the central driver of cellular respiration. It takes acetyl or CoA produced by the oxidation of pyruvate and originally derived from glucose as its starting material and, in a series of redox reactions, harvests much of its bond energy in the form of NADH, 2-FADH2, and ATP molecules. The reduced electron carriers, NADH and 2-FADH2, is generated in the TCA cycle, will pass their electrons into the electron transport chain, and through oxidative phosphorylation will generate most of the ATP produced in the cellular respiration. In eukaryotes, the citric acid cycle or the Krebs cycle takes place in the matrix of the mitochondria, just like the conversion of pyruvate to acetyl or CoA. In prokaryotes, these steps both take place in the cytoplasm. The last part of the pathway reforms the molecule used in the first step. The cycle includes 8 major steps. In the first step, acetyl or CoA combines with oxaloacetate, a 4-carbon molecule, losing the CoA group and forming the 6-carbon molecule citrate. After citrate undergoes a rearrangement step, it undergoes an oxidation reaction, transferring electrons to NAD+ to form NADH and releasing a molecule of carbon dioxide. The 5-carbon molecule left behind then undergoes a second, similar reaction, transferring electrons to NAD+ to form NADH and releasing a carbon dioxide molecule. The 4-carbon molecule remaining then undergoes a series of transformations, in the course of which GDP and inorganic phosphate are converted into GTP, or in some organisms, ADP and inorganic phosphate are converted into ATP. An FAD molecule is reduced to FADH2, and another NAD+ is reduced to NADH. At the end of the series of reactions, the 4-carbon starting molecule, oxaloacetate, is regenerated, allowing the cycle to begin again.
Pawis ang mas oily. Iba yung kanya pawis talaga ng pawis akin oily.
In the first step of the cycle, acetyl or CoA combines with the 4-carbon acceptor molecule, oxaloacetate, to form a 6-carbon molecule called citrate. After a quick rearrangement, this 6-carbon molecule releases 2 of its carbons as carbon dioxide molecules in a pair of similar reactions, producing a molecule of NADH each time. The enzymes that catalyze these reactions are key regulators of the citric acid cycle, speeding it up or slowing it down based on the cell's energy needs.
Where?
The remaining 4-carbon molecule undergoes a series of additional reactions, first making an ATP, or in some cells, a similar molecule called GTP, then reducing the electron carrier, or FAD, to 2-FADH2. This set of reactions regenerates the starting molecule, oxaloacetate, so the cycle can repeat. Overall, one turn of the citric acid cycle releases 2 carbon dioxide molecules and produces 3 NADH, 1 2-FADH2, and 1 ATP. The Krebs cycle goes around twice for each molecule of glucose that enters cellular respiration, because these are 2 pyruvates and thus 2 acetyl or CoA made per glucose.
The last process of the cellular respiration is the electron transport chain. The electron transport chain forms a gradient across the inner mitochondrial membrane, which drives the synthesis of ATP via chemiosmosis.
The electron transport chain is a series of proteins and organic molecules found in the inner membrane of the mitochondria. Electrons are passed from the one member of the transport chain to another in a series of redox reactions. Energy released in these reactions is captured as a protein gradient, which is then used to make ATP process called chemiosmosis. The next step is electron transfer and proton pumping. As electrons are passed down the chain, they move from a higher to a lower energy level, releasing energy. Some of the energy is used to pump H plus ions, moving them out of the matrix and into the intermembrane space. This pumping establishes as electrochemical gradient.
The first step is the delivery of electrons by NADH and FADH. Reduce electron carriers NADH and FADH from other steps of cellular respiration transfer their electrons to molecules near the beginning of the transport chain. In the process, they're turned back into NAD and FAD, which can be reused in other steps of cellular respiration.
The third step is splitting of oxygen to form water. At the end of the electron transport chain, electrons are transferred to molecular oxygen, which splits in half and takes up H to form a water.
That's how the cellular respiration ends.